Every living thing starts as one cell. This cell can divide, forming complex organisms like humans. Cell division is a controlled process where one cell splits into two. These cells grow, divide, and form groups, becoming tissues, organs, and living organisms. For this to work, the cell must coordinate key activities. Cell division must be timed with DNA replication and growth. This ensures genetic material is copied accurately and passed on without errors. Scientists call this sequence of events the cell cycle. It's the cell's program for growth and reproduction, and understanding it is key to understanding life. The cell cycle is a sequence of events where a cell duplicates its genome and divides into two daughter cells. Cell growth happens continuously, but DNA synthesis only occurs during one stage. The cell cycle has two phases, interphase and M-phase. Interphase is the period between cell divisions, where the cell prepares for division. It has three subphases, G1, S, and G2. G1 is the interval between mitosis and DNA replication. The S phase is when DNA replication occurs. G2 is when proteins are synthesized in preparation for mitosis. Some cells, like heart cells, exit the G1 phase and enter a quiescent stage called G0, where they remain active but don't proliferate. The cell cycle has a dramatic period called M phase, or mitosis. This phase reorganizes the cell and divides it equally. It has two main parts: karyokinesis, which divides the nucleus, and cytokinesis, which divides the cytoplasm. Mitosis has four stages: prophase, metaphase, anaphase, and telophase. In these stages, the cell's components change and move. The nuclear envelope disappears and reappears, and chromosomes condense and separate. After mitosis, cytokinesis completes the cell division. In animals, the cell splits in two with a furrow. In plants, a cell plate forms and grows outward. Mitosis is a process found mostly in diploid cells. It's also found in some haploid cells of lower plants and social insects. Mitosis produces daughter cells that are genetically identical to the parent cell. This process is crucial for growth in multicellular organisms as it increases size and complexity. Mitosis also maintains the balance between the nucleus and cytoplasm in cells. It enables cell repair, replacing cells in tissues like the epidermis and gut lining. In plants, mitosis allows certain tissues to divide continuously, enabling ongoing growth and development throughout the plant's life. Meiosis is a specialized cell division that reduces the chromosome number by half. It involves two sequential nuclear and cell divisions, but only one round of DNA replication. This process creates 4 haploid cells. Meiosis is crucial in the life cycle of sexually reproducing organisms, creating the haploid phase. It occurs during gametogenesis in plants and animals, producing haploid gametes. The process has two main parts: meiosis I and meiosis II. Meiosis I is the reduction division, and meiosis II functions like normal mitosis, ultimately producing 4 haploid daughter cells. Meiosis has two key roles in organisms that reproduce sexually. It maintains the chromosome number for each species across generations. Meiosis reduces the chromosome number by half, producing gametes with a haploid set of chromosomes. When gametes fuse during fertilization, the diploid number is restored in the offspring. Meiosis also generates genetic variation within populations. This variation is driven by two mechanisms: crossing over and independent assortment of chromosomes. Crossing over exchanges genetic material between homologous chromosomes. Independent assortment randomly mixes maternal and paternal chromosomes in each gamete. These processes create genetic diversity, which is essential for evolution and adaptation to changing environments. The cell cycle is the sequence of events for cell duplication and division. It includes interphase and M-phase, which is mitosis, the actual cell division. During division, chromosomes duplicate into 2 identical halves called chromatids. These chromatids are joined at the centromere, where spindle fibers attach. Spindle fibers are microtubules that pull and move chromosomes. Division has two parts: karyokinesis, which is nuclear division, and cytokinesis, which is the division of the cytoplasm. In meiosis, homologous chromosomes pair up, forming a bivalent or tetrad. This process is called synapsis. Cell divisions are classified into two types: equational division, which conserves chromosome number, and reduction division, which halves it. Mitosis and meiosis are two different types of cell division. Mitosis occurs in somatic cells, which are regular body cells. Meiosis takes place in germ cells, which become gametes. Mitosis involves one division, while meiosis has two sequential divisions. This affects the chromosome number in the resulting cells. Mitosis produces 2 diploid daughter cells, while meiosis produces 4 haploid cells. Cells from mitosis are genetically identical to the parent cell. Meiosis creates genetic variation through crossing over and synapsis. Mitosis handles growth, tissue repair, and asexual reproduction. Meiosis is for gamete formation, enabling sexual reproduction and genetic diversity. Cell cycles move at different speeds. Human cells take about 24 hours to complete one cycle. Yeast cells take roughly 90 minutes. The duration depends on the organism and cell type. Some cells, like heart muscle cells in animals and nerve cells in mammals, never divide and stay in a resting state. When division happens, mitosis reproduces somatic cells. In animals, mitosis is used for diploid somatic cells. Plants use mitosis for both haploid and diploid cells. Meiosis produces gametes for sexual reproduction in plants and animals. It's also part of alternation of generations in plants, where the organism switches between life stages with different chromosome numbers.